Often a tree that is capable of flowering looks the same as one that cannot flower. Leaves, twigs, phyllotaxy, and bark all seem similar. Ivy is a common plant with distinct juvenile and adult morphology. Juvenile ivy has palmately lobed leaves and climbs. It is the form you see on walls, whereas adult ivy has entire, ovate leaves and upright stems that do not climb. Ivy can persist in the juvenile condition for years, growing extensively over walls and fences, and then individual branches may undergo phase change and start to flower while adjacent branches continue in the non-flowering juvenile phase. Several genera of cacti have a much more pronounced difference between juvenile and adult phases. Juveniles of all species of Melocactus and Bachybergia grow as cacti with ordinary bodies for many years, but when they undergo phase change, the adult shoot has a narrower pith and cortex. The cortex is not green and not photosynthetic. A different kind of wood is produced. Phyllotaxy changes. Epidermis is immediately replaced by bark, and the size, shape, and abundance of spines are different. The adult phase differs so strongly from the juvenile phase that a single plant appears to be two entirely different plants grafted together. In Espistoa and Fachiroa, when a phase change occurs, it affects only one narrow strip of the shoot. As the shoot apical meristem continues to function, one side of the shoot it produces is adult and able to produce flowers, and the other sides of the same shoot are juvenile and cannot flower. Figure 14.27. The green base of this melocactus is its juvenile phase, and the red top is its adult phase. The two are parts of a single shoot produced by just one apical meristem. While young, the plant grew for years as a juvenile, with a body resembling that of many cacti, but it was unable to flower. Once old enough, it underwent phase change and the apical meristem switched to producing the narrower, very spiny adult phase, which can flower. The adult portion cannot photosynthesize because of the dense covering of spines. For the rest of its life, this plant will continue to grow as an adult, with the cephalium becoming taller and the juvenile base remaining as it is. No new photosynthetic tissues will be formed. Photoperiodic induction to flower. The conversion of an adult plant from the vegetative to the flowering condition may be the most complex of all morphogenic processes. This is not one process, different mechanisms exist in different species. In certain annual species, size appears to be the only important factor. Peas and corn initiate flowers automatically after a particular number of leaves has been produced, regardless of environmental conditions. Flowering is controlled by internal mechanisms. In many species, perhaps most, transition to the flowering condition is triggered by photoperiod, day length, which acts as a season indicator. One subclass of these plants blooms when days are short and are short-day plants. Another subclass, long-day plants, is induced to bloom when days are long, in summer. Plants that do not respond to day length are day-neutral plants. Figure 14.28. The green parts of this Espistoa are its juvenile body, and the brown strips are its adult phase. While young, it grew as just a juvenile that had about 20 branches, but as each branch became old enough, each underwent a phase change. Only a strip of each branch became adult, however, the rest of each branch continued to develop with juvenile characters. Flowers are produced only from the brown adult regions. Unlike Melocactus, new photosynthetic tissues are produced every year in Espistoa. Table 14, for photoperiodic species. All plants fall into one of these three photoperiod categories. This table lists only a few familiar examples. We know much about how plants measure day length. First, the pigment phytochrome detects the presence or absence of light. Phytochrome has a light-absorbing portion attached to a small protein of about 125,000 daltons. Phytochrome occurs as dimers in cells. When phytochrome absorbs red light with a wavelength of approximately 660 nanometers, the chromophore changes shape, and this forces the protein to change its folding. This affects many of its properties, one of which is its hydrophobicity. In the refold state, it is more hydrophobic and binds to membranes. A second altered property is its absorption spectrum. It now absorbs not at 660 nanometers, but in the far red region of 730 nanometers. When this form absorbs far red light, however, it refolds back to the red absorbing form and releases from the membrane. The two forms are called P and PFR. Also, PFR reverts to P in darkness. Apparently, P is inactive metabolically but becomes morphogenically active and exerts its effect when it absorbs red light and is converted to PFR. PFR is the active form and brings about metabolic responses. PFR is transported into the nucleus and binds to one or more proteins, and the combination of the two apparently activates particular genes. After a plant is given red light, converting phytochrome to the active PFR form, exposure to far red light converts phytochrome back to the inactive P form. If far red light is given quickly enough after red light, phytochrome does not have enough time to affect cell metabolism and no effect is seen. However, if far red comes long enough after red for the presentation time to be fulfilled, PFR is able to complete the transduction process and far red light can no longer cancel the red light stimulation. Figure 14.29 The chromophore or light-detecting portion of one particular phytochrome. The rest of the molecule is a protein. The two portions are joined by a sulfur atom. We now know that there is a family of at least 5 closely related phytochromes. Some have overlapping functions, some have distinct functions. Many details are being investigated now. Table 14, 5 photomorphogenic responses for which phytochrome is the photoreceptor. A plant experiencing a short day in nature automatically receives a long night. Similarly, long days are always accompanied by short nights. Night length is actually the critical factor. A long day plant is in reality a short night plant. It can be placed in a growth chamber and artificially given both long days and long nights, for example, 16 hours of light and 16 hours of dark in a 32-hour day. If day length is the important factor, the plant should flower because it has long days. If night is the critical factor, then it should not flower because it does not have short nights. When the experiment is done, the long day plant does not flower, indicating that night length is critical, not day length. Similar experiments have shown that short day plants really are long night plants. If given a 16-hour cycle, they do not flower, but if given long nights, even accompanied by artificially long days, the plants are induced to flower. Each species has its own particular requirements for long or short nights, that is, not all long nights have to be the same length. Instead, each species has a critical night length. If a short night plant receives nights shorter than this critical length, it flowers, whereas a long night plant must receive nights longer than its own critical night length. Because critical night length varies from species to species, it is possible for a long night plant and a short night plant to bloom under the same conditions if the critical night length for the short night plant happens to be longer than that for the long night plant. Table 14, 6 concurrent flowering of long day and short day plants. Although Xanthium stromarium is a short day plant and Hyoscyamus niger is a long day plant, both flower if given days 14 to 12 hours long. Those days have nights 10 to 12 hours long, longer than the 8.4 HR critical night length of Xanthium, but shorter than the 13.6 HR critical night length of Hyoscyamus. Day length, night length control of flowering and other processes such as the initiation or breaking of dormancy is more common at locations farther from the equator. Away from the equator, nights become progressively shorter from winter to summer and then progressively longer from summer to winter. The greater the distance from the equator, the greater the length of the longest winter night and the shorter the length of the shortest summer night. Thus, if two species are to bloom just after the beginning of May, a species in the southern United States or Mexico must have a critical night length shorter than that of a species in the northern United States or Canada. Presentation time varies considerably. In some morning glories, one photoperiod of the proper length induces flowering, whereas at least 1 or 2 weeks of proper photoperiods are necessary for other species. The accuracy with which night lengths can be measured varies, but the most accurate species known is the long day plant henbane. It must have nights shorter than 13 hours, 40 minutes. If the nights are even 20 minutes too long, 14 hours long, for example, it does not flower. Figure 14.30 in natural 24-hour light-dark cycles must have short nights any time the days are long and long nights when days are short. Thus, plants could detect season by measuring either day or night. When photoperiodism was first discovered, it was assumed that day length was important, therefore, all our terminology was based on that. With electric lights, it is possible to create a long photoperiod, 32 hours long here, with both long days and long nights. Under these conditions, short day plants bloom, but long day plants do not. When phytochrome was discovered to be responsible for measuring night length, it was hypothesized that most phytochrome was converted to PFR by the end of a day and then reverted slowly back to P during the night. It was postulated that if it could completely revert in the dark, before the next sunrise, metabolic changes would be triggered. However, it is now known that virtually all PFR converts back to P within 3 or 4 hours, a time far too short to be a night measuring clock by itself. It must instead interact with another type of clock that occurs in plant cells. The sites of perception for night length are young leaves. It is possible to stimulate one leaf with a spotlight and induce the plant to flower, even if the apical meristem, the site of response, is not illuminated. Because the site of perception is not the site of response, a chemical messenger must be transmitted between the two. If a leaf is photoinduced and then immediately cut off the plant, no flowering occurs. If it is allowed to remain attached for several hours, the flowering stimulus is synthesized and transported out of the leaf. If the leaf is then removed, the plant still flowers. An obvious experiment is to induce a leaf by giving it the proper night length, then collect the sap that is transported through the petiole and assay it for the hormone that acts on the apical meristem. This has been done hundreds of times by many people, without any repeatable success. The process is not as simple as we had at first thought. An extremely interesting set of results has been obtained by grafting together plants with different photoperiod requirements. Individuals of the tobacco species Nicotiana sylvestris are long-day plants. The species N. tabacum has 2 types of individuals, those of the cultivar N. Tabacum CV travasond are day-neutral plants, and those of N. tabacum CV Maryland mammoth are short-day plants. When long-day and day-neutral plants are grafted together and given long days, the day-neutral plant is also induced to flower, presumably by a floral stimulus that passes through the graft union. Similarly, day-neutral plants are induced to flower if grafted to short-day plants and given short days. Grafting one day-neutral plant to another does not increase flowering, so the grafting by itself has no effect. Figure 14:31. If a short night plant is given long nights, it does not flower, but it is possible to cause flowering by illumination with 15 minutes of dim red light. The plant acts as though it has received 2 short nights separated by a 15-minute day. The red light night break does not have to be given to the entire plant. If a narrow beam of red light shines on a single leaf while all of the rest of the plant remains in darkness, the plant flowers. When the combination of short-day plants grafted to day-neutral plants was given long days, the short-day plants did not flower as expected, and the day-neutral partners flowered about the time they would have if not grafted to anything. When long-day plants grafted to day-neutral plants were exposed to non-inductive short days, however, not only did the long-day plants not flower, but the day-neutral partners were also prevented from flowering. Apparently, under non-inductive conditions, long-day plants actually produce an inhibitor of flowering. Flowering in these long-day tobacco plants seems to be controlled by a switch from inhibitor production to promoter production, whereas in the short-day tobacco plants, flowering is controlled only by the presence or absence of a promoter. Neither the promoter nor the inhibitor has been isolated, but gibberellins are suspected to be involved in the synthesis or activation of the promoter. A new advance in our understanding of the control of flowering is the discovery that when leaves are given the proper day length, a gene called FT is activated. This gene codes for a protein called FT that is carried through phloem to the apical meristems. This is an exciting breakthrough, but control of flowering is complex, and we must anticipate that there are many other steps to be discovered with many variations involving numerous genes other than just FT. Endogenous rhythms and flowering. Plants contain endogenous rhythms, that is, certain aspects of their metabolism cycle repeatedly between 2 states, and the cycle is controlled by internal factors, by an internal clock. The most obvious example of this is in the sleep movements of the leaves of plants like prayer plant. In the evening, leaflets drop down, and in the morning, they raise themselves to the horizontal position as motor cells increase their turgor. It is easy to assume that this is a photonastic response, but if the plants are placed in continuous darkness, the leaflet position continues to change. They return to the up position about every 24 hours and move down about 12 hours later, controlled not by dawn or dusk, but by their own internal clock. In many flowers, the production of nectar and fragrance is also controlled by an endogenous rhythm and occurs periodically even in uniform, extended dark conditions. The underlying mechanism that constitutes the clock is poorly understood but is known to involve a negative feedback loop. Imagine just two proteins, A and B, with A activating B, and with B inhibiting A. As A works, B becomes more active and thus inhibits A, but as A becomes inhibited, it can no longer activate B. Consequently, B's activity lowers, which allows A's activity to increase. The system can cycle rhythmically indefinitely. In Arabidopsis thaliana, the negative feedback loop involves at least 3 genes rather than just 2. TOC1 activate LHY and CCA1, but one of the effects of these latter two is to inhibit TOC1. When placed in uniform conditions, the endogenous cycle typically differs slightly from 24 hours, being either somewhat longer or shorter. However, in nature, the rhythm is exactly 24 hours long because light is able to entrain the rhythm. The pigment responsible for detecting the light for entrainment is phytochrome. Each morning, the red light of sunrise resets the rhythm so it can never get out of synchronization with exogenous light-dark cycles. Many types of endogenous rhythms have a period that is not 24 hours long. Cytoplasmic streaming and the spiraling motion of elongating stem tips have periods of only a few minutes to a few hours. These are ultradian rhythms. If a period is approximately 24 hours long, it is a circadian rhythm, the most common kind. The release of gametes in brown algae is controlled by a 28-day lunar rhythm. Some seeds have an annual rhythm of germinability. If stored in uniform conditions and periodically provided with moisture and warmth, they germinate only at times of the rhythm that correspond to springtime. The involvement of endogenous circadian rhythms in flowering was discovered during dark interruption experiments. A short-day plant can be prevented from flowering by interrupting long nights with a brief exposure of red light. This is detected by phytochrome, and the plant acts as though it has received 2 short nights separated by a 15-minute day. Short-day plants, given a very long night, continuous darkness, have an endogenous rhythm of sensitivity to light breaks. If the light break is given at 6 hours into the dark period, or at 30 hours, 54 hours, and so on, the light break prevents flowering. These times correspond to darkness in a normal environment. But if the light break is given at a time when the endogenous rhythm would be expecting normal daylight conditions, such as at 16, 40, or 64 hours after the beginning of the dark treatment, the light break does not stop flowering. Plants kept in uniform, dark conditions undergo an endogenous cyclic sensitivity and insensitivity to red light interruption of the critical night length. Just how the endogenous rhythm and the critical night length work together to stimulate flowering is not known. Table 14. 7 metabolic processes that undergo endogenous rhythms in plants. A brief description of photoreceptors is presented in Table 14.8. ABC model of flower organization. All flowers share the same basic developmental organization. Most have 4 types of appendages. Outermost are the sepals, then petals, stamens, and finally carpels occur in the center. There are many variations. Wind-pollinated flowers often lack sepals and petals, and in many species certain flowers lack carpels and others lack stamens. Many of the plants we cultivate in flower gardens are double flowers, which means they have extra petals but no stamens. It is typical for eudicot flowers to have 5 of each appendage, 5 sepals, 5 petals, 5 stamens, and 5 carpels, but some flowers have dozens of petals, 100 or more stamens, and numerous carpels. Figure 14.32. Under natural conditions, a circadian rhythm matches the cycle of light and dark, being exactly 24 hours long. In continuous darkness, most circadian rhythms have periods slightly longer than 24 hours. In nature, sunrise resets the clock by acting on phytochrome every morning. Figure 14.33 If a short-day plant is placed in continuous darkness, it will flower. The plant can be prevented from flowering by giving it red light night breaks, but these are effective only if given at those times of the endogenous rhythm when the plant is expecting darkness. If given when the rhythm is expecting light, the light breaks are ineffective and the plant will bloom. Whatever metabolism red light interrupts does not occur continuously in prolonged darkness, but rather periodically, controlled by the internal clock. Despite the diversity, it appears as if the basic organization of most flowers can be explained by a hypothesis called the ABC model. This model postulates that three fundamental genes, A, B, and C, interact to control the basic aspects of flower organization. Gene A affects the outermost, lowest regions of a flower shoot apical meristem, gene C controls the uppermost, central region, and gene B partially overlaps the other two. In the outermost, lowest, donut-shaped region where only gene A is expressed, the primordia develop into a whorl of sepals. Primordia in the next region upward and inward are influenced by both genes A and B, and they develop as petals. The combination of genes B and C produces stamens, and in the very center of the flower where only gene C is expressed, primordia develop as carpels. Table 14.8 Photoreceptors. Figure 14.34 This wild rose has only a few petals and is a single flower. Many cultivated roses have been bred to be double flowers, with a mutation that causes them to produce extra petals in the place of stamens. Figure 14.35 This Graptopetalum is the type of flower often thought of as being a typical eudicot flower. It has 5 sepals, 5 petals, 5 stamens, and 5 carpels. This flower of epiphyllum differs from that of a typical eudicot by having dozens of petals, almost 100 stamens, and 11 carpels. Whereas each floral apical meristem of most species produces only a few primordia, those of cacti produce hundreds. This model was proposed and is supported by many types of aberrant flowers. For example, if gene B is mutated and non-functional, sepals are produced in the outer part of the flower and carpels occupy the inner parts. There are no petals or stamens. More importantly, when these flowers are examined closely, they have an outer whorl of sepals, as is normal, and then an inner whorl of sepals where we would expect petals. Then there is a whorl of carpels where we would expect stamens. Finally, there is the innermost set of normal carpels. Although such a flower has just 2 types of appendages, they are still arranged in 4 whorls. The important thing is that normal appendages are produced in the wrong regions. A mutation that causes an organ to develop in an unexpected site is called a homeotic mutation. Mutations in gene A result in carpels in whorl 1 and stamens in whorl 2. The ABC model has been so successful that it is now the basis for numerous experiments. Many plant geneticists induce mutations in well-studied species such as Arabidopsis thaliana and Antirrhinum majus, searching for more details about the ways in which these genes are controlled and how they work together. Because there are so many types of flowers, we expect to discover not only many versions of A, B, and C but also many other genes that subtly or dramatically influence A, B, and C. An example of modified flowers whose development still is unknown is those that consist of just stamens or carpels without any other appendages. Figure 14.36 The ABC model proposes that the outer part of a floral apical meristem is controlled by gene A, the inner part by gene C, and gene B overlaps the other two. Notice the outermost area is controlled only by gene A and produces sepals, the innermost is controlled only by gene C and produces carpels. Petals are produced by the zone where both A and B are active, and stamens occur where both B and C are active. If a mutation inactivates gene B, then the floral apex has only 2 zones, the one controlled by gene A produces 2 whorls of sepals, and the one controlled by gene C produces 2 whorls of carpels. If gene B is inactivated, then the floral apex does not produce either petals or stamens. Figure 14.37 Each cluster on this slender, pendant stalk is an oak flower, consisting of just stamens. Elsewhere on the oak tree are flowers that consist of just carpels. Cattails produce two types of extremely reduced flowers. At the top are thousands of flowers that consist of just stamens, and at the bottom are flowers that consist of just carpels. Both types of flower are extremely simple, consisting mostly of either stamens or carpels without sepals or petals. A few hairs may be present. In this photograph, the carpellate flowers have been pollinated and have developed into fruits. You are unlikely to ever see carpellate flowers of cattails unless you dissect them from the plant before they emerge above the leaves. Plants and people. Box 14-3 Environmental Stimuli and Global Climate Change. Global climate change is causing our world to rapidly become warmer and wetter, but plant mechanisms for detecting and responding to environmental stimuli are changing more slowly, if at all. As we burn oil, coal, and natural gas, and as we convert forests into pasture for cattle, we increase the amount of greenhouse gases in the atmosphere, causing the air, soil, lakes, and oceans to become warmer. As ocean temperatures rise, their surface waters evaporate faster, making the atmosphere more humid and increasing the amount of rain and snow that later fall on land. Temperatures do not increase uniformly everywhere. Instead, circulation patterns in the atmosphere and oceans are affected, so some areas become warmer, others cooler, some wetter, others drier. Changing climate will have profound effects on all plants, not only on those that respond to temperatures, but also on those controlled by day length. Increasing temperatures affect two critically important events for temperate plants. The date of the last frost in spring occurs earlier, and the time of the first frost in autumn comes later. The frost-free growing season in many areas starts earlier and ends later. Plants have a longer growing season. Plants that germinate or bud out solely based on temperatures can take advantage of this longer growing period, and many seem to be thriving. But for plants controlled by photoperiod, their critical night length does not change. They germinate or bud out at the same time in spring as they have for centuries, and they go dormant at their typical time in autumn. They are not able to take advantage of the extra days of warmth in spring and autumn. Instead, they are dormant when they could be photosynthesizing, growing, and reproducing. And, just as bad, their respiration during dormancy is higher than before because it is controlled by environmental temperature. Not only are the plants not producing sugars photosynthetically as long as they could, they are now respiring away their carbohydrates faster. They will have less reserve nutrients available when they resume growth in springtime. Now consider the interaction of photoperiodic plants and temperature-controlled plants. They occur together in the same habitat and compete with each other for water, minerals, room for their roots, and so on. As warm temperatures occur earlier in spring, the temperature-controlled plants get a head start over the photoperiod-controlled ones, and the same is true in autumn. It is likely that the photoperiod-controlled plants will suffer in this competition, and the ratio of the two types of plants in the ecosystem will change. As temperature in general increases, the snow-free habitable zone in alpine areas gradually rises to higher elevations. Similarly, habitable zones are expanding northward in the northern hemisphere, southward in the southern. Areas near the north and south poles are more hospitable. Again, temperature-controlled plants may benefit from this. If their seeds happen to occur in the newly warmer areas, they should be able to grow and reproduce. But the same is only partially true for photoperiod-controlled plants. These should be able to grow higher on any mountain on which they exist already. The critical night length is the same up and down the mountain. But close to the poles, a few days at the beginning of summer have sunlight for 24 hours. There is no night at all for a few days. And at the beginning of winter, several days have no sunlight. From early winter to early summer, day length increases from 0.0 to 24 hours. At the equator, daylight always lasts for 12 hours and night is also 12 hours, all year long. Between these two extremes, days get slightly longer each day in lower latitudes and much longer each day in high latitudes. For plants that need very long days to bloom, that occurs in May or June in the northern part of the United States and southern Canada, but it occurs in March in northern Alaska and Canada. If a longer growing season would allow that species to grow that far north, its critical night length would occur too early while the plant is still a seedling. It could grow in the new habitat but not reproduce there. It is important to remember that plants and their control mechanisms do evolve. Hundreds of different plant species differ in their critical night length, and this variation came about through evolution by natural selection. The important question is whether these mechanisms will evolve rapidly enough to allow plants to adapt to the changing climate. We do not know the answer, but, in general, such evolution is slow, and we are causing the climate to change rapidly. At the next level. 1. Regulatory cascades. When a hormone binds to its receptor, the first reaction is the activation of one or several enzymes, often by the addition or removal of a phosphate group. These enzymes activate others, which in turn activate others. Through the control cascade, one event, hormone binding, affects many different aspects of cell development. We know a great deal about control cascades, and there is much more that we do not know. This is a complex topic for independent study, so it is best to find one particular hormone you are interested in, or one particular development pathway, and focus only on those. 2. Perception of environmental signals. After many years of frustration, we are finally constructing a clear, unambiguous model of many types of signal perception in plants. Light signals have been useful because we can experimentally control color and intensity very precisely, and we know that light must be detected by a pigment. I recommend light perception as a topic for independent study, but any aspect of signal perception will be rewarding. Just keep in mind that this is a very active area of research and many gaps remain, as there are many experiments in which the results are not well understood. 3. The Arabidopsis book. The American Society of Plant Biologists provides a wonderful free service to all botanists and other biologists by maintaining an online compilation of information about the model organism Arabidopsis thaliana. This has illustrations, reviews, summaries, essays, and original research articles. It is updated regularly, so it is worthwhile to visit the site frequently. 4. Control of the initiation of flowering. For almost a century, the term florigen was used as a code word for the unknown flowering messenger. It was known that for plants whose flowering is controlled by day length, the stimulus had to be given to a leaf, but the response would occur by inducing shoot apical meristems to change from leaf to flower production. It was assumed that florigen was a mixture of chemicals because no single chemical could be extracted from induced leaves that would have the desired effect when applied to a shoot apical meristem. In the last several years, it has finally been discovered that the messenger RNA for a gene called flowering locus is either the flowering signal or the dominant part of a complex of several chemicals. Because this is a crucial step in reproduction, it is well worth independent study. Summary. 1. It is selectively advantageous for organisms to be able to sense and respond to significant aspects and changes in their environments. 2. Communication between body parts of an organism is essential to the integration and coordination of the organism's metabolism and development. Certain parts may need to respond to environmental or metabolic changes that they cannot sense themselves. 3. Plants must perceive important environmental information, transduce it to a communicable form, and respond to the transduced information. 4. For ways in which plants respond to stimuli are tropic responses, nastic responses, morphogenic responses, and taxes. 5. In general, sites of perception are different from sites of response and must be linked by a means of communication. Presentation time and threshold are important elements in perception. Most communication appears to be by hormones. 6. Currently, the known classes of plant hormones are auxins, cytokinins, gibberellins, abscisic acid, ethylene, jasmonic acid, and several others. 7. The response to a hormone depends on which hormone is acting, the preparation of the responding cell, and the simultaneous or sequential presence of other hormones. 8. Flowering may involve the following steps: competence to be induced, occurrence of inductive conditions, sufficient health to produce flowers, and later stimuli to induce flowers to open. 9. Flowering and many other seasonal responses are controlled by night length in many species. Phytochrome is involved in measuring night length. 10. Plants contain endogenous rhythms, cyclic changes in their metabolism. The rhythms most frequently are circadian, having a period of approximately 24 hours. These rhythms affect numerous aspects of plant metabolism. Important terms: signal amplification, cascade statocytes, statolith symmetry, taxis. Threshold transduction of a stimulus, tropic response, vernalization. Review questions. 1. Most plants are so large their bodies exist in several different environments. What does this mean? Give examples for a single plant. 2. For a stimulus to cause a plant to change its metabolism or development, there must be 3 actions, and 3. Before a plant can respond to environmental factors, they must be perceived. Give examples of several environmental cues that plants perceive. 4. After a plant perceives a stimulus and before it can respond, there is another step called. What happens in this step? 5. Name 5 environmental factors that plants detect as stimuli. In each case, what types of information are provided to the plant? 6. Different prefixes are used for various stimuli. What is the prefix for each of the following? A. Light. B. Gravity. C. Touch. D. Chemical. 7. How does a positive tropic response differ from a negative tropic response? What is a diatropic response? 8. How does a nastic response differ from a tropic response? What is a morphogenic response? 9. In the perception of a stimulus, what are presentation time and threshold? 10. Responses can be either of 2 types and 11. What is an all or none response? Give some examples of plants that flower with an all or none response. 12. What is a dosage-dependent response? Name some plants that flower this way. 13. What are the 4 ways that a plant can respond to a stimulus? Define and give examples of each. Why are some responses classified as positive, negative, or plagio-, whereas others are not? 14. Almost all plant communication is by a slow mechanism. What is the mechanism? 15. Name the known classes of plant hormones. What are the characteristics of plant hormones? 16. Name 1 natural auxin. 17. The concentration of substances as powerful as hormones can be controlled by 3 methods in plants. What are the 3 methods? 18. Auxin, like many substances, can be transported through the phloem. However, there is a second mechanism that exists just for auxin transport. What is it? 19. Like auxin, cytokinins are involved in dozens of responses in all parts of the plant. Name 1 important response. 20. About how many gibberellins are known to exist? Are all of them active hormones? 21. What is the only gaseous hormone? How is it involved in fruit ripening? 22. What is apical dominance? How is auxin involved? 23. Describe the mechanism by which auxin appears to control leaf abscission. 24. Bending of plant parts toward or away from stimuli requires. 25. Studies of phototropism often use which part of an oat seedling? 26. True or false, despite years of intensive research, we still do not have even an outline of the mechanism that actually causes plants to begin producing flowers. 27. Almost all plants must reach a certain age before they can be induced to flower. Before that point, they are said to be in the stage. Afterward, when they can be induced, they are in the stage. 28. Light is often the most important environmental factor for inducing a plant to flower, but it is not the intensity of light. What is the important aspect? 29. What are long-day plants? Short-day plants? Day-neutral plants? What is the critical night length? 30. Which pigment is responsible for detecting presence or absence of light? What are its 2 forms? 31. We know that short-day plants do not really need short days, but actually long nights. Describe the type of treatment that proves this. Is some kind of light given during the night or some period of darkness given during the day? 32. What is the difference between endogenous and exogenous rhythms? Give several examples of each type, and be certain to include several that have either short periods or long ones. 33. List 8 metabolic processes that undergo endogenous rhythms in plants. 34. If an endogenous rhythm has a period of about 24 hours long, we say it is a rhythm. 35. Imagine a short-day plant being given continuous darkness. If it is given a flash of red light at a time when its endogenous rhythm would be expecting daylight, will that red light inhibit flowering? What about if the red light is given at a time when the plant's endogenous rhythm is expecting a long night? Design credits: Hummingbird, Tango 58 Moment Getty, Green Plant Cells, Shutterstock Incorporated Natalia Ora, Purple Tulip, Shutterstock Incorporated Maurice C. Fields, Dandelion, Shutterstock Incorporated Daniel Kreisel, Poppy, Shutterstock Incorporated Saryuri, Plant Icon, Shutterstock Incorporated Vector, Digging Man Icon, Shutterstock Incorporated ZArt. Chapter 15. Genes and the Genetic Basis of Metabolism and Development. Chapter opener image: each cell in this leaf of corn contains the same genes as every other cell here, and that is true of the genes in the nuclei, mitochondria, and plastids. Despite having the same genes, various cells develop particular characters and metabolisms because they activate certain genes and repress others. For example, cells in the epidermis express the genes that guide the formation of cutin and wax, but other cells repress these genes. Cells in the xylem activate lignin genes, but other cells do not. Outline. Concepts. Storing genetic information. Protecting the genes. The genetic code. The structure of genes. Transcription of genes. Protein synthesis. Ribosomes. tRNA. mRNA translation. Control of protein levels. Analysis of genes and recombinant DNA techniques. Nucleic acid hybridization. Restriction endonucleases. Identifying DNA fragments. DNA cloning. DNA sequencing. Sequencing entire genomes. Genetic engineering of plants. Viruses. Virus structure. Virus metabolism. Formation of new virus particles. Origin of viruses. Plant diseases caused by viruses. Box 15-1 Plants and People, Genetic Engineering and Evolution. Learning objectives. After reading this chapter, students will be able to explain the differential activation of genes. Recall the 3 ways DNA is safely and accurately stored. Summarize plant gene structure and the transcription process. Discuss the function of ribosomes and tRNA. Summarize mRNA translation from initiation through termination. Restate the process through which transcription factors control gene activity. Describe the different methods of gene analysis. List 1 use for DNA cloning. Recall 2 methods of DNA sequencing. State 1 difficulty in plant genetic engineering. Discuss the process of using viruses as vectors in plant genetic engineering. Did you know? Our genes control our development partly by directing the synthesis of enzymes such that needed metabolic pathways are functional. Other genes direct the production of structural proteins that alter cell shape. Very few genes code for only one character, such as brown eyes versus blue eyes. Most genes affect parts of one or several metabolic pathways, so when they mutate, they affect multiple pathways and characters. Concepts. Plants are composed of numerous types of cells. Each cell type is unique because it has a distinct metabolism based largely on proteins such as enzymes, microtubules, and membrane proteins. Although all cells carry out a fundamental metabolism involving respiration, amino acid synthesis, and so on. Some of the reactions in each cell type differ from those in other cell types. Each may have one or more characteristic types of enzymes or other proteins. For example, enzymes involved in synthesis of flower color pigments are present in petal cells, but not in cells of roots, wood, and bark. Also, sclerenchyma cells contain all enzymes necessary for producing and lignifying secondary walls, but these enzymes and metabolic pathways are not present in parenchyma cells. Cells also differ in shape, again largely owing to differences in their proteins. All tracheids and vessel elements probably have the same enzymes and metabolism for secondary wall deposition, but the pattern of wall deposition varies, guided by a pattern of protein microtubules in the protoplasm. Similarly, cell divisions occur in precise patterns, and as a result, there must be an underlying pattern in the cell that causes the mitotic spindle to have the proper alignment. The information needed to construct each type of protein is stored in genes, but because an organism grows by mitosis, duplication division, all of its cells have identical sets of genes. As each cell differentiates and develops a unique suite of proteins, the underlying developmental process is the differential activation of genes. In the maturing epidermis, genes that code for cutin synthesizing enzymes are turned on, whereas in other cells they remain turned off. On the other hand, genes for P protein remain quiescent in all except phloem cells. Studies of development and morphogenesis examine the mechanisms by which some genes are activated and others are repressed. Figure 15. One cell differentiation is controlled by regulating particular genes in each type of cell. These petals have enzymes necessary for synthesis of pigments. These enzymes are not produced in most other cells of the plant, although all cells contain the necessary genes. These wood cells had many enzymes not found in the petal cells of the same plant. During differentiation, each cell type became unique as different genes were activated, different proteins were produced, and their metabolisms diverged into unique pathways. Chlorenchyma cells differ from other types by having well-developed chloroplasts. These cells have differentiated such that starch storage and release are the dominant aspects of metabolism. They probably have no unique enzymes. All cells can metabolize starch. Degree of activity of a particular set of proteins is important in differentiation. These vessels and fibers have similar, if not identical, metabolisms for synthesis and lignification of walls. They differ primarily in cell shape and pattern of secondary wall deposition. Precise positioning of cellular elements is also critical during differentiation. Table 15. 1. Nucleotides. Nucleic acids contain 5 bases, abbreviated T, A, G, C, and U. One base plus a 5-carbon sugar is a nucleoside, and with a phosphate attached, a nucleoside becomes a nucleotide. Base plus sugar equals nucleoside. Base plus sugar plus phosphate equals nucleotide. When DNA is synthesized, deoxyribonucleotides, those that contain the sugar deoxyribose, are used. When RNA is made, ribonucleotides, containing ribose, are used. Uracil does not occur in DNA, and thymine is absent from RNA. Figure 15. 2. Because all body cells are produced by mitosis, they all have the same genes. Certain basic metabolism genes are probably active in all cells, but specific pathways become active during differentiation, probably because the genes that code for the enzymes of those pathways become active. Once fully mature, the cells may go back to basal metabolism. Epidermal cells often produce cutin only when differentiating, not after maturity. Sieve elements lose their nuclei as part of maturation, and tracheary elements digest away all of their protoplasm. They have no metabolism at all when mature. During protein synthesis, the correct amino acids must be incorporated in the proper sequence because this determines both the structure and all other properties of the protein. The cell must contain a source of information that holds the sequence information for all its proteins. This information archive is DNA, deoxyribonucleic acid. DNA is a linear, unbranched polymer composed of 4 types of deoxynucleotide monomers, usually abbreviated A, T, G, and C. Once actually polymerized into DNA, the base portion of each nucleotide monomer protrudes as a side group. It is the sequence of nucleotide side groups that is the information needed to synthesize proteins correctly. A gene is each region of DNA that is responsible for coding the amino acid sequence in a particular protein. Each type of protein has its own gene. Both environment and protoplasm also contain information vitally important for plant growth, morphogenesis, and survival. The environment provides informative cues about season, moisture availability, time for seed germination, time for flowering, and direction of gravity. These environmental and metabolic signals must be converted into chemical messengers that enter the nucleus and interact with genes. If the signals indicate that the cell is to differentiate into a vessel element, all genes that produce enzymes necessary for synthesis and lignification of a secondary wall must be located and activated. Genes that code for proteins that guide a particular pattern of wall deposition also must be turned on. Conversely, the cell must be inhibited from undergoing any further cell division. Genes involved in mitosis and cytokinesis must be repressed. Several techniques permit botanists to locate the genes for many proteins. The genes can then be isolated in vitro, duplicated, and their nucleotide sequences revealed. Currently, our knowledge is still limited, but these techniques of DNA sequence analysis are so powerful that progress is extremely rapid. Similar techniques make it possible to alter the DNA sequence and then insert the gene back into a plant cell. As the cell grows, divides, and differentiates, the altered DNA either produces an altered protein if the coding region was changed or produces the protein at an unusual time or place if its control site was changed. These recombinant DNA techniques, sometimes called genetic engineering, are helping us understand the processes that occur between the perception of a stimulus and the plant's response to that stimulus. In addition, recombinant DNA techniques permit us to change features of plants, for example, making them more resistant to insects or having seeds and fruits that are more nutritious for us. Storing genetic information. Protecting the genes. It is critically important that the information in DNA be stored accurately for a long time. If storage is not safe, the information produced by the DNA will be inaccurate and probably useless or even harmful. There are several ways in which DNA is kept relatively inert and safely stored. 1. DNA does not participate directly in protein synthesis. Instead, DNA produces a messenger molecule, messenger RNA, which carries information from DNA to the site of protein synthesis. The mRNA, not DNA, is exposed to the numerous enzymes, substrates, activators, and controlling factors of protein synthesis. If mRNA is damaged, it can be replaced with more copies of mRNA. Within a single cell, thousands of individual molecules of a particular enzyme may be needed. If the DNA itself had to direct the synthesis of each protein molecule, it would probably be damaged long before enough protein had been synthesized. Instead, however, DNA directs the production of several copies of mRNA, each of which directs the production of hundreds of protein molecules. 2. Most DNA is stored in the nucleus, protected from the cytoplasm by the nuclear envelope. During interphase, the nuclear envelope forms the outer boundary of the nucleus, keeping most cytoplasmic components out and the proper nuclear substances in. The DNA of plastids and mitochondria is protected from cytosol enzymes by being located within plastids and mitochondria themselves. Nuclear genes in plants store information for approximately 20,000 to 40,000 types of proteins. Plastid DNA encodes only about 50 to 100 genes, and mitochondrial DNA specifies less than 40. 3. Histone proteins hold most nuclear DNA in an inert, resistant form. Histones are a special class of proteins found in all organisms that have nuclei. There are 5 types: H1, H2A, H2B, H3, and H4. The last 4 are among the most highly conserved proteins known, that is, the sequence of amino acids in the histones of one organism is virtually identical to the sequence in any other organism. For example, the H4 histone contains 103 amino acids, and its sequence in higher animals, such as cows, differs from its sequence in higher plants, such as peas, at only 2 sites. Histone proteins are so essential that virtually any change in their amino acid sequence causes the organism to die or at least not reproduce. Figure 15.3 When a gene is active, its sequence of nucleotides guides the synthesis of heterogeneous nuclear RNA, which is modified into messenger RNA and transported to the cytoplasm. mRNA binds to ribosomes that translate its nucleotide sequence and polymerize amino acids in the proper order, thus creating a protein. Ribosomes on the left have just started, thus, their proteins are still short. Ribosomes on the right have read almost all the RNA, so their proteins are longer, almost complete. Histones form aggregates and DNA wraps around them, forming a spherical structure called a nucleosome. Histone H1 then binds nucleosomes into a tightly coiled configuration. In this mode, the DNA protein structure, chromatin, is so dense that enzymes cannot penetrate it, and DNA is relatively inert. Even if it is exposed directly to DNA-digesting enzymes, called DNases, histone-bound DNA is not extensively damaged. However, chromatin is still sensitive to regulatory molecules and can be unpacked in preparation for synthesis of mRNA or for replication of DNA during the S phase of the cell cycle. Figure 15 for the nucleotide sequence for the gene for histone H4 in wheat is presented in the top row of each set of lines. In the second row is the sequence for the same gene for a different type of wheat. Where the two genes are identical, only a dash appears for the second gene. The portion of the gene that codes for protein begins in the fifth line, and the amino acid sequence, the primary structure of the protein, is given in the third row of each set of lines. Wherever a mutation has caused the second gene to code for an amino acid different from the first gene, that amino acid is given in the fourth row. Although 18 mutations have occurred, the resulting amino acid sequence is unchanged except at one site. The non-coding regions of the gene, from the beginning to +1 and from +309 to the end, are not highly conserved, and the two genes differ greatly in these sites. Plastids, mitochondria, and prokaryotes have no histones or nucleosomes, their DNA is naked. The genetic code. 20 types of amino acids are used in synthesizing proteins, but only 4 different nucleotides are present in DNA or mRNA. Consequently, it is not possible for one nucleotide alone to specify one amino acid because 16 amino acids would be left without nucleotides to code for them. Similarly, nucleotides cannot be used simply in pairs of 2, such as AU for isoleucine or CC for proline, because there are only 16 possible pairs. It is necessary for nucleotides to be read and used in groups of 3. 64 possible triplets, known as codons, can be made using 4 nucleotides. Table 15.2 shows which amino acid is coded by each codon. Notice that codon refers to triplets in mRNA, not in DNA. With 64 possible triplets, a surplus of 44 codons remains after each amino acid is paired with a codon. 3 codons, UA, UAG, and UGA, are stop codons. They signal that the ribosome should stop protein synthesis. August is the start codon that signals the point in mRNA where protein synthesis should begin. The extra 40 codons also code for amino acids, so most amino acids have 2 or more codons. For example, both UUU and UUC code for phenylalanine, and CAU and CAC code for histidine. Because multiple codons exist for most amino acids, the genetic code is said to be degenerate. Degeneracy further protects DNA. A mutation in DNA might change a codon in mRNA from UUU to UUC, for example, but because both code for phenylalanine, the same protein is produced before and after this particular mutation. The genetic code is almost perfectly universal, all organisms and genetic systems but one share the genetic code shown in Table 15. 2 viruses, prokaryotes, fungi, animals, and plants all use the same codons to specify particular amino acids, and the same is true for plastid DNA. Only in mitochondria are several codons changed. This almost universal commonality of the genetic code is one of the strongest pieces of evidence that life arose only once on Earth and that all living organisms have evolved from one ancestral organism. The structure of genes. Most genes, up to 90% in any cell, are quiescent most of the time and are activated and read only when the cell needs the particular enzymes they code for. Each gene must have a structure that allows controlling substances to recognize the gene, bind to it, and activate it at the proper time. Table 15, 2. The codons of mRNA. The triplets in mRNA are codons. The DNA triplets in the gene itself are their complements. The first base of the codon is listed on the left and the second base at the top. Within each large box are the 4 possible third bases and the amino acids specified by each. Genes are composed of a structural region that actually codes for the amino acid sequence, and a promoter, a controlling region involved in regulating the synthesis of mRNA from the structural region. The promoter is located upstream from the structural region, that is, to the 5-foot side. It varies in length from gene to gene but can be several hundred nucleotides long. Certain regions are particularly important. One, called the TATA box, is a short sequence about 6 to 8 base pairs long rich in A and T. If the TATA box is damaged by either mutation or experimental treatment, the RNA synthesizing enzyme RNA polymerase II does not bind well. Most eukaryotic genes have other promoter sequences called enhancer elements located even farther upstream, as many as several hundred base pairs away from the structural region of the gene. When a hormone alters cell metabolism, it does so by producing intracellular chemical messengers that activate genes either by binding directly with the promoter region or by binding with proteins that then interact with the promoter. After activating agents have bound to the promoter, RNA polymerase II can attach. Figure 15, 5 carbons in ribose and deoxyribose are numbered from 1 foot to 5, with phosphate attached to the 5 carbon and the hydroxyl used in polymerization attached to the 3 carbon. Any nucleic acid has a 5-foot end and a 3 end. A gene is always written with the 5-foot end, and thus the promoter, on the left. Nucleotides are numbered beginning at the left boundary of the structural region. Nucleotides to the left of this are given negative numbers and are said to be upstream. Because DNA is double-stranded, its length is measured as the number of nucleotide pairs or base pairs, whereas RNA, being single-stranded, is measured as the number of nucleotides or bases. After RNA polymerase II binds to the promoter, it migrates downstream toward the structural region. However, it does not create any RNA until it is approximately 20 to 30 nucleotides below the TATA box. The RNA polymerase might be expected to search for the DNA equivalent of the August start codon of mRNA, but that is not the case. If some of the DNA is artificially removed between the TATA box and the normal start site, the RNA polymerase begins synthesizing RNA farther downstream than normal. Like those of prokaryotes, genes of plastids and mitochondria often have multiple promoters, not just one. Plastids use 2 types of RNA polymerase, one imported from the nucleus and one they make themselves, and which is remarkably similar to that of the bacterium Escherichia coli. The structural portion of genes contains 2 distinct types of regions, exons and introns. Exons are sequences of nucleotides whose codons are eventually expressed as sequences of amino acids in proteins, and introns are sequences of nucleotides that are not expressed, but instead intervene between exons. Several plant genes have just 2 or 3 introns, the gene for Ruby P. carboxylase and the genes for the storage proteins glycinin and phasolin of legume cotyledons. The gene that codes for the protein portion of phytochrome has 5 introns, one of which is 1,500 base pairs long. Genes with 25 introns occur, as do genes with no introns. It is very common for exons to make up much less than half the structural part of a gene. Figure 15, 6 ways of illustrating introns and exons in maps of genes. The gene for leghemoglobin in legumes has 3 introns. Intron I occurs between bases 32 and 33 of the finished mRNA, intron 2 between bases 68 and 69, and so on. 2 genes in corn produce 2 similar enzymes, both called alcohol dehydrogenase and distinguished as ADH1 and ADH2. The numbers above and below the triangles indicate the number of bases present in each intron. The 2 genes are similar, having 9 introns located at the same positions. Corresponding exons in ADH1 and ADH2 are the same length, but corresponding introns may be quite different. Intron 1 is 534 bases long in ADH1 but only 95 bases long in ADH2. Transcription of genes. After RNA polymerase binds and encounters the start signal, it begins actually creating RNA, a process called transcription. The two strands of DNA separate from each other over a short distance, and free ribonucleotides diffuse to the region. If a ribonucleotide containing cytosine approaches a DNA nucleotide that contains guanine, the two form 3 hydrogen bonds and remain together, at least temporarily. Wherever DNA contains T, a free A can form 2 hydrogen bonds to it. Similarly, free U forms 2 hydrogen bonds with A in DNA, and so on with G and C. RNA polymerase binds the free ribonucleotide, holds it, and catalyzes the formation of a covalent bond, forming RNA. As each covalent bond is formed, 2 high-energy phosphate bonding orbitals are broken. Polymerization is thus a highly exergonic process that cannot be easily reversed. Free ribonucleotides diffuse to and pair with both strands of DNA, but RNA polymerase is located only on the strand that has the gene. The complementary strand is not read. Transcription proceeds rapidly, incorporating about 30 ribonucleotides per second, with the DNA double helix unwinding ahead of the moving enzyme. After RNA polymerase moves off the promoter initiation site, a new molecule of RNA polymerase binds and begins synthesizing another molecule of RNA. Whereas 2 molecules of DNA wrap around each other into a double helix, RNA-DNA duplexes do not. The RNA polymer that emerges from RNA polymerase releases from the DNA. Figure 15.7 When a gene is turned on, the DNA double helix separates over a short region. Free ribonucleotides diffuse in and pair with the region of temporarily single-stranded DNA. The formation of 2 or 3 hydrogen bonds between the DNA deoxyribonucleotides and the free ribonucleotides allows the DNA sequence to control the sequence of the RNA being formed. RNA polymerase transcribes both introns and exons into a large molecule of hnRNA that is rapidly modified by nuclear enzymes. Introns are recognized, cut out, and degraded back to free ribonucleotides. Exons are spliced together, resulting in an RNA molecule, all of which codes for amino acids. All RNA destined to become mRNA is somehow recognized by an enzyme that binds to it and attaches a series of adenosine ribonucleotides on its 3-foot end, forming a polytail approximately 200 bases long, the only exception being the mRNAs for histone proteins. Another step in message processing involves changing the first nucleotide into 7-methylguanosine. Ultimately, a completed mRNA is produced and transported from nucleus to cytoplasm. RNA polymerase continues to act until it encounters a transcription stop signal in the DNA. The stop signal of several genes consists of 2 parts. The first is a short series of DNA nucleotides that are self-complementary, that is, the RNA transcribed from them can double back on itself and hydrogen bond to another part of itself. This results in a small kink, a hairpin loop that is believed to affect RNA polymerase. Just downstream of this region of DNA is a long series of adenines. Various protein factors also are involved. In their absence, RNA polymerase sometimes continues transcribing, reading the next region of DNA as if it were also part of the gene. Plastid and mitochondrial genomes have prokaryotic organization. Their genes are not separated by long stretches of space or DNA, but instead genes occur in sets, one following another immediately. One set of promoters causes RNA polymerase to transcribe not just a single gene but actually several before it releases. This long, multi-gene transcript is then processed differently than is hnRNA. Figure 15.8 DNA can be carefully extracted from a nucleus, allowed to spread out, and then prepared for examination in an electron microscope. These are ribosomal genes being transcribed into long RNAs that will later be cut into 3 separate rRNAs. The diagram explains each type of line. Figure 15.9 The primary transcript, hnRNA, has its introns cleaved out and the exons spliced together. The introns are depolymerized back to free nucleotides. An enzyme adds up to 200 adenosine ribonucleotides to the end of the RNA. These are not coded by thymidines in the DNA. The first nucleotide at the 5-foot end is converted to 7-methylguanosine. Protein synthesis. In the process of protein synthesis, ribosomes bind to mRNA and read its codons. Guided by the information in the nucleotide sequence of the mRNA, the ribosomes catalyze the polymerization of amino acids in the order specified by the gene from which the mRNA was transcribed. Ribosomes. Ribosomes are small particles that read the genetic message in mRNA and construct proteins guided by that information. Each is composed of two subunits, one larger than the other, and each is made up of both proteins and ribosomal RNA. The small subunit contains one molecule of rRNA, the large subunit contains one molecule each of three types of rRNA. The number of proteins present in eukaryotic ribosomes is known to be greater than 80. Ribosomes found in the cytoplasm of eukaryotes are designated ADS, meaning that they are relatively large and dense. Ribosomes of plastids, mitochondria, and prokaryotes are smaller, lighter 70S ribosomes. The nuclear genes for the three largest rRNAs of ADS ribosomes are unusual because they occur tightly grouped together and act as a single gene with just one promoter. Transcription of this cluster, which is located in the nucleolus, produces a long RNA molecule that is then cut into three pieces. Short regions are digested off the ends of these pieces, resulting in three rRNA molecules. The gene for the smallest rRNA molecule is not located in the nucleolus but out in the chromatin along with protein-coding genes. These genes are transcribed into rRNA, not mRNA. They do not code for proteins. Table 15. 3 components of ADS cytoplasmic ribosomes. Once transcribed, rRNA molecules combine with ribosome proteins in the nucleolus, forming one large particle that then is cleaved into the large and small subunits of a ribosome. These subunits are then transported out of the nucleolus through the nucleus to the cytoplasm. All cells need large numbers of ribosomes. Most genes are present in a diploid nucleus as only 2 copies, but just 2 copies would be inadequate for producing the hundreds of thousands of rRNAs needed. Instead, rRNA genes are highly amplified, that is, many copies of each are present. In flax plants, each nucleus may have up to 120,000 copies of the gene for the small rRNA and 2,700 copies of the gene cluster for the 3 large rRNA molecules. Figure 15. 10 ribosomes have 2 subunits, one large and one small. When they fit together, there is a groove for mRNA to pass through, a channel through which the growing protein emerges, and a channel into which amino acid carriers enter. Figure 15. 11. The genes for the 18S, 5.8S, and 28S ribosomal RNAs occur together as a cluster, separated by short regions of space or DNA. All 3 genes are transcribed as just one molecule of RNA, spacers included. Spacer RNA is cut out, resulting in 3 individual rRNA molecules. Upstream from the 18S gene is the promoter, and then a region made up of 10 to 15 copies of a short sequence about 135 base pairs long, called subrepeats. They are not transcribed. On either side of the rRNA gene cluster are more rRNA clusters, as many as 2,700, all occurring end-to-end as a gene family. KB is kilobases, 1 kilobit equals 1,000 bases. tRNA. During protein synthesis, amino acids are carried to ribosomes by ribonucleic acids called transfer RNA. tRNAs are necessary because a codon cannot interact directly with an amino acid. The genetic code can be read only by a ribonucleic acid that has a 3-nucleotide sequence, called an anticodon, that is complementary to and hydrogen bonds to the codon. For example, UUU and UUC are both codons for phenylalanine. The tRNAs that carry phenylalanine have the complementary anticodons of either AAA or GA. GAA looks backward for something that must bind to UUC. By convention, nucleotide sequences are written 5'3", which is GA. An alternative is to write the numbers to show it as being written backward for easier comparison with the codon, 3'AG5. There are as many types of tRNA as there are codons that specify amino acids. Stop codons do not have tRNAs. All tRNAs have the same parts, an anticodon and an amino acid attachment site at its 3' end consisting of the sequence CCA. A special class of enzymes recognizes each tRNA and attaches the correct amino acid to it. This step, called amino acid activation, must be precise. If the wrong amino acid is placed onto a tRNA, it becomes incorporated into the protein as if it were the correct amino acid, causing the protein to have an erroneous structure. Transfer RNAs contain bases not found in other nucleotides. tRNAs are transcribed from tRNA genes, and like all ribonucleic acids, they contain the common bases A, U, G, and C. However, after these are polymerized into tRNA, enzymes modify some bases to unusual forms. Each tRNA carries an amino acid only briefly. After it is activated, it rapidly encounters a ribosome that is reading the codon complementary to its anticodon. It gives up its amino acid and shuttles back to the cytosol and is reactivated. Even though each tRNA can cycle like this many times per second, millions of tRNAs are needed in every cell, and hundreds or even thousands of copies of each tRNA gene may be present in each nucleus. The nucleus contains genes for all 61 types of cytoplasmic tRNA, but plant mitochondria have genes for only a few tRNAs and must import the rest from the cytoplasm. Plastids have genes for only about 30 types of tRNA. mRNA translation. Initiation of translation. The synthesis of a protein molecule by ribosomes under the guidance of mRNA is called translation. Protein synthesis begins with a complex initiation process involving the start codon August. This codes for the amino acid methionine, but two types of tRNA actually carry methionine, and they have different properties. One, called initiator tRNA, binds to the ribosome small subunit even before any mRNA is present. Several initiation factors, mostly proteins called eukaryotic initiation factors, also bind to the small subunit. In this condition, the complex is competent to bind to mRNA, after which the large subunit of the ribosome binds to the complex and the initiation factors are released. Just how mRNA is recognized is not known, but the initiator tRNA is important for finding the August start codon and positioning the small subunit. Figure 15:12 and all tRNAs have this general shape, with at least 3 loops caused by self-complementary base pairing. The small fourth loop is present only in some. The anticodon is at the middle loop and the amino acid is attached to the CCA at the 3' end. This is one of the 6 tRNAs that carry leucine. It has the anticodon CA, which recognizes the codon 3' GUU5, normally written UUG. The unusual symbols indicate bases that are chemically modified into unusual forms. tRNA folds into an L shape rather than lying flat. Figure 15:13 Protein synthesis. The steps are explained in the text. It is critically important that the ribosome be properly aligned on mRNA because the sequence of nucleotides can be read in any set of 3. The sequence CUGCAG can be read as CUUGCACAG and would code for leucine alanine glutamine, but if the ribosome binds incorrectly, shifted downstream by one nucleotide, the mRNA is read as CUUGCACAG, the codons for phenylalanine histidine and either serine or arginine, depending on the next nucleotide. Reading nucleotides in the wrong sets of 3 is a frameshift error, because virtually all codons are misread. Frameshift errors typically result in completely useless proteins. Elongation of the protein chain. mRNA lies in a channel between the 2 ribosome subunits. Extending outward from the mRNA channel are 2 grooves, each wide enough for a tRNA to fit into it, such that the tRNA anticodon can touch an mRNA codon. When both channels contain activated tRNAs, adjacent codons are being read. Plants and people. Box 15-1 Genetic Engineering and Evolution. An important area of genetic engineering is the production of herbicide-resistant crop plants. The concept is to engineer plants that are not harmed by an herbicide, such that fields of the plant can be sprayed with the herbicide to kill weeds without harming the crop itself. There are arguments for and against the basic idea of using herbicides rather than using organic gardening, but for the moment, let us consider just the genetic engineering aspect. An extremely effective herbicide called glyphosate became available in the 1970s. Glyphosate has many favorable features. First, it inhibits an enzyme necessary for the synthesis of 3 aromatic amino acids. This alone would make it deadly by blocking protein synthesis due to missing amino acids. In addition, however, these amino acids themselves are used in other essential plant pathways, and thus, glyphosate is especially lethal. Second, the enzyme that it inhibits occurs only in plants. Animals do not have this enzyme. So being exposed to glyphosate does not harm animals. Third, glyphosate rarely pollutes water because it binds so strongly to soil that it does not wash into groundwater or streams. In fact, it is applied by spraying it onto leaves rather than mixing it with soil. Finally, it breaks down quickly into harmless products. The one drawback of glyphosate is that it kills all plants. It can be applied to fields only before the crop seeds germinate, killing weeds that germinated before the crop did, or if crop plants are tall and weeds are short, it can be sprayed below the crop's leaves and onto the weed's leaves. Genetic engineering entered the picture when it was discovered that bacteria have a gene called CP4 that synthesizes the 3 amino acids but is immune to glyphosate. Plant scientists isolated the gene and genetically engineered soybeans to use this gene in addition to the natural plant gene. These soybeans would not die if sprayed with glyphosate. An entire field of these soybeans could be sprayed at any time to kill weeds without harming the crop. Currently, glyphosate-resistant alfalfa, canola, cotton, corn, and sugar beets have been genetically engineered. This is so effective that many farmers have stopped using other herbicides and rely on glyphosate. An added benefit is that they do not have to plow their fields before planting. Plowing inhibits weeds by burying their seeds deeply. However, it loosens soil so much that it greatly increases erosion, and it requires large amounts of fuel. The extensive use of a potent herbicide like this creates a strong selection pressure for resistance among other plants. Any weed that has a mutation that makes it resistant to glyphosate will thrive in a field with fertilizer, irrigation, pesticides, and no competition from other weeds. People thought it would be almost impossible for resistance to evolve because the plant enzyme is so fundamental. How could an entire new enzyme or whole new pathway evolve in a short period of time? But glyphosate-resistant weeds have appeared already in many parts of the world, and not because they have a new enzyme. Instead, plants are variable in their capacity to transport glyphosate through phloem. Most transport glyphosate throughout their body, and it accumulates in shoot and root tips, stopping growth. But a small number of plants have something different about their phloem that causes them to transport glyphosate to leaf tips. It harms the leaf tips, but does not hurt the rest of the plant. In natural environments, this unusual transport may not help the plant at all, but it is extremely beneficial in a field being sprayed with glyphosate. Glyphosate did not cause glyphosate resistance to come into existence. This feature would have been present already in a naturally variable population of plants. It is just that these plants suddenly are more adapted because glyphosate is being used in their environment. They will become a greater part of their population as susceptible plants are killed. Other types of resistance should be expected. It may be that plants already exist that have degrading enzymes that can break glyphosate down or that have membranes that are impermeable to it. The continued use of glyphosate will give these plants a selective advantage, and their numbers will increase. It is a simple, clear-cut case of evolution by natural selection. This experience should remind us that plants and evolution can respond in numerous ways, many of which we might not anticipate. Other areas in which GM crops are being produced and where we might want to be cautious are drought resistance and insect resistance. Plants have already been genetically engineered to require less water for their survival. They require less irrigation, so less water can be diverted from rivers and lakes, which would then retain more of their natural condition. Would seeds of drought-resistant crop plants then be able to invade natural, dry areas and become invasive pests on their own, crowding out the natural plants? A potent insect-killing protein is produced by the bacterium Bacillus thuringiensis. It is easy to transfer that gene into plants, creating plants that resist insects. The gene, however, is also expressed in pollen of genetically modified plants, and poisonous pollen blows away, especially from cornfields, landing on the leaves of other plants, coating them with a poisonous dust that could harm any kind of insect. As is so often the case, advances in technology bring with them both benefits and risks. By analyzing each opportunity for its merits and dangers, we should be able to increase our quality of life without harming the environment, but we must always remember that we are dealing with organisms that can evolve and respond to the altered conditions we are creating. Figure 15:14. Because there are no spacers between codons, the ribonucleotides can be read in three possible ways. Each results in completely different proteins, only one of which has the proper primary structure. At the time of initial binding, the small subunit already contains initiator tRNA in the P channel. The adjacent A channel is empty, but numerous molecules enter it at random. Some are activated tRNAs, but if their anticodons do not complement the exposed codon at the bottom of the A channel, they diffuse out. If a tRNA with the proper, complementary anticodon enters, hydrogen bonds form between the codon and the anticodon. This holds the tRNA in place long enough for an even more stable binding to occur. Enzymes located in the large ribosomal subunit break the bond between the methionine and its tRNA in the P channel, simultaneously attaching the methionine to the amino group of the amino acid on the tRNA in the A channel. This reaction needs no outside source of power. The broken bond is a high-energy bond, whereas the newly formed peptide bond is only a low-energy bond. The empty tRNA, freed of its methionine, is released from the P channel, and the ribosome pulls itself along the mRNA for a distance of 3 nucleotides. This movement is powered by GTP and seems to be performed by proteins on the large subunit. As the ribosome slides along the mRNA, the A channel slides to the next codon, and the tRNA with the 2 amino acids attached becomes surrounded by the P channel, not the A channel. When the proper activated tRNA diffuses into the A channel and hydrogen bonding between codon and anticodon occurs, the large subunit enzymes again release the short protein chain from the tRNA in the P channel and attach it to the amino acid on the tRNA in the A channel, creating a protein 3 amino acids long. This process repeats until the ribosome reaches a stop codon. Termination of translation. When a stop codon is pulled into the A channel, normal elongation cannot occur. No tRNA is present with an anticodon complementary to a stop codon. Instead, a release factor enters the channel and stimulates the large subunit enzymes to initiate the normal reactions. The high-energy bond holding the protein to the P channel tRNA is broken, and a new bond is formed with water, releasing the protein from both the tRNA and the ribosome. The tRNA is released, and the small subunit dissociates from the large subunit and releases the mRNA. All components diffuse away, but as soon as the small subunit encounters an initiator tRNA and other initiating factors, it binds to another mRNA and the process begins again. In summary, protein synthesis involves the following steps. RNA polymerase transcribes hnRNA, being guided by the sequence of DNA nucleotides in a gene. The hnRNA is processed into mRNA as introns are cut out and exons are spliced together. After moving from nucleus to cytoplasm, mRNA binds with ribosomes, and tRNAs carry amino acids to the ribosome mRNA complex. The ribosome translates the mRNA codon by codon, and tRNAs fit into the ribosome only when their anticodon is complementary to the codon in the A channel. After the protein is completed, it is released and the ribosome subunits detach from the mRNA. All components diffuse away from each other, but the ribosome subunits can translate the next mRNA they happen to meet. The mRNA can be translated again by other ribosomes. Control of protein levels. As cells undergo differentiation and morphogenesis, their metabolism and structure become different from those of other cells because of the presence of proteins, especially enzymes, unique to that cell type. A central question in developmental biology is the mechanism by which distinct cell types control the activities of genes so that they undergo the proper differentiation and obtain the proper set of proteins. There are several points at which protein synthesis and activity theoretically could be controlled, making a gene physically available for transcription, nature of the promoter region, processing of hnRNA into mRNA, transport of mRNA from nucleus to cytoplasm, binding of mRNA to the ribosome small subunit, rate of translation, processing of protein, and activation or inactivation of the protein. Many enzymes and structural proteins are present in a cell in an inactive form. Tubulin is an excellent example. A cell may contain a large pool of tubulin monomers and then aggregate them into microtubules at a specific time. Microtubules can appear rapidly without the need for gene activation or protein synthesis. Similarly, enzymes are often completely inactive until phosphate groups are added by a class of enzymes, called kinases, that are themselves activated by the arrival of hormones. Hormones arrive at the plasma membrane and bind to receptors, which then synthesize second messengers that enter the cell. These activate the phosphorylating enzymes that in turn activate the dormant enzymes, leading to a significant change in the cell's metabolism. This is mostly an activating rather than a differentiating mechanism. These cells are already prepared for highly specific responses. It would be extremely inefficient if a cell transcribed all its genes and synthesized all of its possible proteins when only a few are needed. For maximum efficiency, a cell should not even synthesize the mRNA for proteins it does not need. We expect that the most fundamental level of control of morphogenesis would occur at the level of transcription. We know very little about control of transcription in eukaryotes, especially plants, but our knowledge is increasing rapidly. In many cases, gene activity is controlled by transcription factors, proteins that bind to promoter or enhancer regions and activate genes. Many transcription factors have a sequence of amino acids that fits into the large groove of a DNA double helix, recognizing a specific sequence of nucleotide bases. Interestingly, most transcription factors must act as dimers. Two similar factors first recognize each other, and only then can they bind to the proper promoter. By acting as dimers, a moderate number of transcription factors can control a very large number of genes. For example, 3 proteins A, B, and C can form 6 dimers, A, AB, AC, BB, BC, and CC. The binding of transcription factors to DNA creates a structure to which RNA polymerase can bind. Many transcription factors can be classified together based on their overall structure. Helix-turn helix transcription factors have 2 regions of alpha helix joined by a looping section of protein in basic leucine zipper transcription factors. The 2 parts of a dimer are held together by interactions of leucine amino acids on their surfaces, and so on. Because transcription factors come from somewhere else and bind to DNA, they are said to be transacting factors. Promoters, enhancers, and tautoboxes are part of the gene itself and thus are cis-acting factors. Gene expression is also controlled by a family of short RNA molecules called microRNAs. Many types of microRNAs are now known, and there is a large number of names as well. However, each name ends in RNA, so they are easy to recognize when you read about them. These short pieces of RNA act in many ways, some recognizing sites on DNA itself and binding there, and others recognizing sites on hnRNA or mRNA and attaching to those molecules. In many cases, binding by microRNAs inhibits gene expression or mRNA translation. Because RNA consists of nucleotides, it has the potential to recognize sequences in DNA and RNA very precisely and thus exert precise control over gene action. Analysis of genes and recombinant DNA techniques. Nucleic acid hybridization. The 2 halves of a DNA double helix can be separated by heating them just enough to break the hydrogen bonds between complementary bases. This separation, which produces a solution of single-stranded DNA molecules, is called both DNA melting and DNA denaturation. If the solution of single-stranded DNAs is cooled slowly to a temperature low enough for hydrogen bonds to reform, 2 molecules with complementary sequences form hydrogen bonds and stick together whenever they collide. If the 2 halves of one piece encounter each other, all of their bases pair and adhere firmly even at a relatively high temperature, but just by chance, many pieces have short sequences that are complementary, whereas most of their sequences are not. If these encounter each other, they form only a small number of hydrogen bonds and probably fall apart again if the mixture is cooling only slowly. As the degree of complementarity decreases, the stability of pairing decreases. The reformation of double-stranded DNA by cooling a solution of single-stranded DNAs, called both DNA hybridization and reannealing, is used to determine the relatedness of 2 types of DNA. For example, DNA can be extracted from 2 organisms, cut into small pieces, and melted. the sample from one is attached to a filter, and the other sample, which has been made radioactive, is poured over it at a temperature that permits hybridization. If the two organisms are related, a large amount of the radioactive DNA hydrogen bonds firmly to the DNA on the filter, so the filter becomes very radioactive. If the two species are not closely related, they have so few sequences in common that few hydrogen bonds form, and most of the radioactive DNA pours through, therefore, the filter does not become radioactive. This method is also used to measure the number of copies of a gene that occurs in a nucleus. It had always been assumed that any diploid nucleus contains two copies of every gene, one from the paternal parent and one on the homologous chromosome inherited from the maternal parent. If such DNA is broken into small pieces, denatured, and allowed to cool, the reannealing is extremely slow. Of the hundreds of thousands of pieces, each has only two that can pair with it, its own that had melted away from it and one from the homologous chromosome. Thus, each piece undergoes thousands of collisions before its complement is encountered and stable reannealing occurs. Although most genes do act this way and reannealing is extremely slow, a portion reanneals very rapidly, seeming to encounter complementary pieces easily, as if thousands of the same gene occur. These are genes for rRNA and tRNA, which have thousands of copies in each nucleus. Restriction endonucleases. Natural DNA is such a long molecule it cannot be worked with easily. It can be broken into smaller pieces by chemical treatment or simply by agitating a solution violently. When breaking it into fragments of a more manageable size, it is often critical to cut it at specific known sites so that repeat experiments can yield the same pieces as the first experiment. Before the 1970s, this was impossible. Both chemical treatment and agitation cut DNA at random, and no two experiments ever yielded the same pieces of DNA. Then a class of bacterial enzymes, restriction endonucleases, was discovered. Each restriction endonuclease recognizes and binds to a specific sequence of nucleotides in DNA and then cleaves the DNA. Because of these properties, we always know exactly where DNA will be cut by a particular restriction endonuclease, and when two identical batches of DNA are treated with the same restriction endonuclease, the resulting fragments are always the same. Figure 15 in mild heating of DNA disrupts hydrogen bonding, allowing the two halves to separate into single-stranded molecules. And cooling allows them to reanneal. And in the millions of nucleotides in a single DNA molecule, short sequences occur many times simply by chance, so almost any two pieces have a few short complementary sequences. However, much of the sequences are mismatched and do not adhere. The poorly bonded DNA duplexes fall apart if the solution is still warm, giving each piece another chance to encounter its true complementary partner. If two identical pieces of DNA are broken at random, each produces pieces perfectly complementary to those produced by the other, but because the pieces are unequal in length, a large amount of single-stranded DNA remains after allowing them to reanneal. The sequence recognized by a restriction endonuclease is present in both strands, running in opposite directions. Such sequences are palindromes. The sequence can be read forward or backward, depending on the strand. Also, because most restriction endonucleases cut each DNA strand near the ends of the palindrome, the two cuts are not aligned. Each end is complementary to and capable of pairing with any other end made by that type of restriction endonuclease. The ends are said to be sticky. Currently, more than 3,000 restriction endonucleases have been discovered, and 600 are commercially available, giving us a large choice of cleavage sites. Table 15 for sites recognized by restriction endonucleases. All fragments produced by a particular class of restriction endonucleases have exactly the same sequence in their single-stranded ends. Thus, if fragments made from the DNA of one organism are mixed with those of another organism, they adhere to each other. A DNA repair enzyme, DNA ligase, can be added to the mixture to repair the cuts so that the two fragments join together. DNA prepared by this method is recombinant DNA. Figure 16. Most restriction endonucleases cut near the ends of palindromes, resulting in pieces with sticky ends. DNA pieces from different sources can be mixed together and tend to adhere, making it much simpler to work with them. both pieces 1 and 3, even though from different organisms, have the same sequence at their ends because BAM-H1, like all restriction endonucleases, binds to and cuts the DNA only if it finds a particular sequence. The same is true of pieces 2 and 4. Identifying DNA fragments evolutionary studies. After restriction endonucleases have acted, the DNA fragments can be identified and used. Most simply, fragments are used directly to study the evolution of DNA. For example, plastid DNA is a small molecule containing only 60,000 to 80,000 base pairs. Treatment with the PSTI restriction endonuclease produces about 10 to 20 fragments. These can be separated by gel electrophoresis and then made visible by staining, producing a restriction map of the plastid DNA. The number of fragments reveals the number of PSTI sites and the number of base pairs between them in the plastid DNA. If plastid DNA is extracted from the chloroplasts of a second, closely related species, it should have the same number of PSTI sites and the same spacing between them as in the first species. If two species are not closely related, their fragment profiles differ. There is a restriction fragment length polymorphism. A mutation may have altered one of the PSTI sites so that PSTI neither recognizes nor binds to it. If so, one less fragment is present, and one of the remaining fragments is extra long. Conversely, a mutation may have altered an ordinary sequence, converting it to a new PSTI site and thus producing an extra fragment. In addition, two species may have equal numbers of fragments, but with a fragment that is longer in one species than the corresponding fragment in the other, a mutation has added extra base pairs to one species or removed some from the other. RFLP analysis is easy, quick, and inexpensive, and it can be done with various restriction endonucleases, each providing its own information about how much change has occurred in the DNA. Figure 15:17 plastid DNA from species A can be cut with a particular restriction endonuclease, resulting in several pieces that can be identified by gel electrophoresis. The enzyme digest is placed on a gel slab and a voltage is applied. Phosphate groups on the nucleotides cause the pieces to move in the electrical field, and the shortest DNA pieces slip quickly through the gel matrix, thus moving the farthest. The largest DNA pieces can barely move through the gel, therefore, they stay at the top. After staining, the gel reveals the number and length of pieces. Here, we have indicated which band on the gel corresponds to each position in the plastid DNA circle, but you would not know that from this type of experiment. In a closely related hypothetical species B, a mutation has changed a nucleotide between A and B so that region does not have the proper sequence for the enzyme, so no cutting occurs. Electrophoresis shows that the two species have 3 bands in common and that species B has 1 band as long as the combined length of the missing 2. For further analysis, some or all fragments can be sequenced, that is, the identity of every nucleotide can be revealed in order by techniques described below. Similar analyses can be performed with mitochondrial DNA, but mitochondria are used more often in studies of animals and fungi. For plant studies, plastid RFLP analysis is more convenient. Nuclear DNA cannot be used easily because there is too much of it, it produces thousands of fragments, and the addition or loss of a few cannot be detected. Physiological studies. In many cases, the objective of the experiment is to locate and isolate one particular fragment, such as the one that contains a specific gene. The following method can be used if any cell forms large quantities of the protein. For example, developing cotyledons of legumes such as beans and soybeans produce large amounts of the storage proteins phaseolin and glycinin, respectively. While the cells are actively synthesizing the protein, ribosomes can be extracted from them and mRNA obtained. The mRNA can be mixed with reverse transcriptase, a virus enzyme that synthesizes DNA using RNA as a template. This complementary DNA is complementary to the exons of the gene. It is synthesized using fluorescent nucleotides. It can then be placed on the various DNA fragments produced by the restriction endonuclease treatment of nuclear chromatin. The cDNA forms hydrogen bonds to whichever fragment contains the gene, and this bonding can be detected by assaying for the fluorescence. The gene itself may contain a restriction site so that it is cut in two and separated into two fragments. The fluorescent cDNA probe hybridizes with both fragments. If this happens, the experiment may have to be repeated with other restriction enzymes that do not cleave inside the gene itself. The best enzyme can be found only by trial and error. Few cells in plants, other than those in cotyledons, produce large amounts of just one protein. As a result, most cells contain hundreds of types of mRNAs, and it is not possible to know which one codes for the protein of interest. Reserve proteins of seeds, however, are some of our most significant, important foods, and hundreds of billions of dollars are spent every year buying storage proteins in the form of beans, wheat, corn, and soybeans. These significant proteins are the objects of intensive study and genetic manipulation. A second method can be used if mRNA is difficult to obtain, but something is known about the amino acid sequence in the protein, even just a little of it. If the sequence of 5 to 10 amino acids is known, the genetic code in Table 15(2) can be used to guess the probable sequence of nucleotides in the corresponding exon. For example, if the known protein sequence contains phenylalanine, the exon may contain either UUU or UUC. After 2 or 3 probable nucleotide sequences have been chosen, they can be synthesized by machines that produce DNA in the form of a radioactive probe. This is used to detect which fragment contains the complementary sequence. Figure 15(18) The method for isolating a particular piece of DNA that contains a gene of interest. T indicates fluorescent thymidine. The gel slab contains fragments made from restriction endonuclease treatment of nuclear DNA. The method is described in the text. If neither of these approaches is possible, all of the restriction endonuclease fragments from the plant are modified and mixed with bacteria. The bacteria take up the fragments, some take up many, others absorb one, and some take up none. In many cases, the plant DNA is digested by the bacterium's own restriction endonucleases, but often it is incorporated into the bacterium's DNA and replicated. The bacteria are spread over dozens of petri dishes, and every bacterium grows into a small colony. Each colony is tested to see if it is making the protein of interest. Any colony that is producing the protein must be replicating the gene as well as transcribing and translating it. That colony can be transferred to a new petri dish and grown. Then DNA is isolated from some of its members and is treated with the same restriction endonuclease that was used to obtain the original plant fragments. At least one fragment of the bacterial digest should match in size one fragment of the plant digest. That fragment contains the gene. It is not possible to compare each of the dozens of bacterial fragments with the tens of thousands of fragments from the original plant material, but that is no longer a problem. Genetically modified bacteria are used, and we know every type of fragment that the bacterial DNA will be digested into when treated with any restriction endonuclease if it has not picked up foreign DNA from our experiment. Any fragment that does not match the maps of the bacterial DNA must correspond to the plant fragment that was introduced. Expression profiling uses cDNAs to examine gene expression during development or to compare development in one species with that in another. For example, all mRNA can be extracted from a young leaf primordium and converted to cDNA, then each type of cDNA is separated from the others. Each type is attached to many glass microscope slides in an orderly matrix, a DNA microarray. Each slide will have thousands of microscopic dots, each dot being a different cDNA and each slide being identical to all other slides. Then mRNA is extracted from an older leaf primordium and placed onto one of the slides such that each mRNA can find and bind to the spot that has its cDNA. Excess mRNA is washed off, and chemicals are added to detect DNA-RNA pairs. Then the slide is examined to see which spots have picked up mRNA. Spots that have no mRNA bound to them correspond to genes that became inactive as the leaf developed from the younger to the older stage. Similarly, mRNA from flower petal primordia might be added to another slide to see which genes are active in both leaf and flower primordia. This does not tell us what the genes are or even their sequence, but by examining microarrays, we can study general gene expression patterns. Figure 15(19) DNA microarray. See text for details. DNA cloning. The method of placing DNA fragments into bacteria, as just described, is an extremely useful technique of DNA cloning. The colony that contains the important fragment can be subcultured and grown easily, and each time a bacterium divides, a new copy of the experimental DNA fragment is made. The bacteria can be cooled, induced to become dormant, and stored for years. When the gene is needed again, the bacteria are revived and cultured. As long as the proper restriction endonuclease is used, the gene can be obtained easily. Placing the original plant DNA fragments into bacteria is much easier than it may seem. Fragments are not simply mixed with bacteria, but are typically combined with plasmids or virus DNA. A plasmid is a short, circular piece of DNA that occurs in bacteria and acts like a tiny bacterial chromosome. Plasmids can move from one bacterium to another and are readily taken up from solution by bacteria. Recall that many restriction endonucleases cut DNA near the ends of a palindrome, resulting in sticky ends. If the plasmid or virus DNA is cut with the same enzyme used to isolate the DNA fragment from the plant, then when the two batches of DNA are mixed, some of the plant DNA adheres to plasmid DNA by means of their complementary ends. DNA ligase is then added to bond the pieces covalently into one continuous double helix. Several plasmids, such as PBR322, have been genetically engineered to be ideal DNA fragment vectors. In their current highly modified form, they have factors that allow them to enter bacteria readily and be replicated. They have antibiotic resistance genes, and they have single sites for several restriction endonucleases so that fragments can be incorporated in precisely known positions. After the fragment is added, the plasmid is mixed with bacteria and absorbed. After a few hours, all bacteria are treated with the appropriate antibiotics. Those that did not absorb a plasmid die. The survivors have the plasmid and therefore DNA fragments from the original plant digest. Figure 15.20 The method by which a plasmid is used to insert plant DNA into a bacterium for cloning. A virus could be used instead of a plasmid. The method is described in the text. Several viruses are being used like plasmids. They contain a short fragment of DNA and can infect bacteria and then replicate. As with plasmids, certain viruses have been modified into highly efficient vectors for genetic engineering. Genetically modified bacteria are easy to culture and handle in the laboratory, but because their genomes are short circles, they cannot accept long fragments of DNA for cloning. Most plant chromosomes are hundreds of times larger than a typical bacterium, and thus, even a short chromosome from a plant nucleus would have to be cut into thousands of fragments for cloning in bacteria. Yeast artificial chromosomes can be used instead. A YAC contains the essential parts of a yeast chromosome plus a gene for drug resistance and parts of plasmid that can accept experimental DNA cut with the proper restriction endonuclease. Such a YAC is tiny and thus has room for a very large amount of DNA, up to 1 million base pairs, to be cloned. After the experimental DNA is inserted, the YAC is placed into a yeast cell, which is much larger than a bacterial cell and can hold more DNA. The yeast cell is then cultured and allowed to grow and multiply. Every time the yeast cells replicate their own chromosomes, they also replicate the YAC. The yeast can be cultured until billions of cells are present, and then some can be used to obtain the cloned DNA. The rest can be frozen indefinitely for use later. The polymerase chain reaction. A powerful technique for DNA cloning is the polymerase chain reaction, in which only enzymes, not living bacteria or yeasts, are used. The sequence to be amplified is heated to separate the two strands of the helix. After cooling, DNA polymerase is added, and it replicates both strands of DNA. This enzyme can add nucleotides only to a pre-existing nucleic acid. Therefore, before the replication can begin, it is necessary to add two types of primer DNA, each complementary to a short region at either end of the sequence to be cloned. End sequences are known if closely related genes are being studied, but often no part of the nucleotide order is known. In that case, artificial DNA, usually a short sequence such as AIA, is added chemically to both ends, then the primer TTTTT will be effective. After the primer has hydrogen bonded to the sequence, a molecule of DNA polymerase attaches and begins working toward the other end. After replication is completed, the mixture is heated temporarily to separate the two strands, then the process is repeated. A heat-stable enzyme, Taq polymerase extracted from hot springs bacteria, is used because it is not denatured by the heating. After each heating replication cycle, there are twice as many copies of the sequence being amplified. Consequently, extremely small amounts of DNA can be cloned very rapidly, and PCR is used for rare copies of DNA. Examples are small amounts of cDNA made from the mRNA of just a few cells, as well as the DNA present in the early stages of viral infection. Or the DNA present in a hair, a drop of blood, or a bit of skin at the scene of a crime. DNA sequencing. Various methods are currently used to sequence DNA. In the chain termination method, DNA to be sequenced is first cloned to obtain a large sample and is then divided into 4 batches. To each batch are added all the enzymes and free nucleotides necessary to carry out DNA duplication. To one tube, a small amount of dideoxyadenosine is also added. A dideoxynucleotide can be added to a growing DNA, however, it cannot react any further, and the growth of the DNA stops. Nucleotides cannot be added to it. In this tube with dideoxyadenosine, the DNA acts as a template and replication begins. When AT is reached in the template molecules, a few incorporate a dideoxyadenosine and stop. Most growing DNA strands incorporate a normal A and keep on growing, then a few stop at the next E, a few more stop at the next, and so on. When the reaction is complete, the test tube contains thousands of DNA molecules of hundreds of sizes, but each size corresponds to the point where AT occurred in the template DNA being analyzed. Similarly, the second test tube contains a small amount of dideoxy T, the third dideoxy C, and the fourth dideoxy G, figure 15 to 21. Until the last few years, the method of analyzing the results was as follows. When all 4 batches are finished, they are loaded into separate lanes in a gel electrophoresis apparatus and allowed to separate, as in Figure 15:17 and Figure 15:22. Each lane contains bands corresponding to the sizes of the DNA molecule. For example, if the DNA contained T as the 3rd, 7th, 8th, and 15th base, the lane containing dideoxyadenosine has bands corresponding to DNAs that are 3, 7, 8, and 15 nucleotides long. The DNA sequence can be read immediately. At present, gels are almost never used. Instead, all processing of DNA samples is now done by machines. Sequences are read and transferred directly to databases. At present, gel electrophoresis has been replaced with much faster methods. In the pyrosequencing method, DNA is added to a solution with all enzymes for replication. In addition, there are other enzymes that release light when pyrophosphate is present. Then a single nucleotide is added. If it is the correct nucleotide, DNA polymerase incorporates it and releases pyrophosphate, which causes a flash of light. The sequencing machine records the nucleotide that caused the flash and then introduces another nucleotide to continue the process. When the wrong nucleotide is introduced, there is no flash, and the sequencer notes that, then washes it away and introduces another. The process is repeated thousands of times as the template DNA is replicated. The latest technology is HiSeq X10, produced by the company Illumina, which became available in 2015. It reportedly can sequence an entire human genome in just 3 days for as little as $1,000. Figure 15:22 diagram of a gel for a DNA fragment. Each lane marked A, T, C, or G corresponds to one of the test tubes. In Figure 15:21 you can read the nucleotide sequence, starting at the top with the shortest, fastest nucleotide. A real gel would be much longer and have hundreds of bands. Figure 15:21 chain termination method of sequencing. Into 4 test tubes, identical pieces of DNA have been placed, all received everything necessary, but each received a small amount of chain terminator. The one depicted here received dideoxyadenosine. If too much of the chain terminator is added, almost all stop at the first T. If too little is added, almost no chains stop at any T, and most finish the entire template. The correct amount produces a good number of copies of every possible length. After incubation, the contents of each test tube would be placed into a lane of a gel electrophoresis machine and separated by length, as shown in Figure 15.22. Computer programs analyze the sequences, searching for regions that might be promoters or enhancers, Tata boxes, August start sites, and areas that might be boundaries between exons and introns. If a region is found that appears to have these gene-like features, it is referred to as an open reading frame. To determine whether it actually is a gene, databases are searched to see if the ORF matches the sequence of any known genes. If there is no matching sequence, it continues to be called an ORF. Often there will be some sequence similarity with a known gene in the same or another organism. If the similarity is high, this ORF is probably a gene with the same function as the known gene, but if the similarity is lower, we are less certain. This may be a real gene, a mutated gene,